Hi, I'm Elizabeth Ellis. I am a Society of Urologic Oncology Fellow at MD Anderson Cancer Center. And today I'm going to be talking about our research in chromophobe renal cell carcinoma. This work was actually funded by the 2024 Chromophobe Award given by the KCA. And so we're really excited to talk to you guys about that today. So as we have talked a lot about in this meeting, sarcomatoid variants of chromophobe RCC are much more aggressive than just general chromophobe RCC. And so this chromophobe RCC sarcomatoid variant is characterized by having TP53 mutations. And specifically we found that when TP53 is mutated, there is a protein called TRP13 that is much more highly expressed. And basically what that means is that it just makes more protein. Of this, much more of this TRP13 protein. And we'll talk a little bit later about how this TRP13 protein works, but essentially it's involved in mitosis, and that means cell division. And so it's allowing those chromosomes to align in order for the cell to then divide. What we did is whole exome sequencing, and we collaborated with Tempus AI, and they have 168 chromophobe renal cell carcinoma patients, and essentially they found that this TP53 mutation, when present, had much higher protein manufacturing of this TRP13 protein. And so then we looked at that in our actual preclinical models, and so we have tissue that we took from a patient tumor, and we grew that in a mouse, and this is called a PDX model, a patient-derived xenograft. And essentially this, once you take the patient tumor and convert it, this actually turned into a sarcomatoid variant, and it's actually showing that this sarcomatoid variant is also making much more of that TRP13 protein. Then we actually look, and you can, it's well known that chromophobe renal cell carcinoma has what we call loss of chromosomes, meaning that the chromosomes aren't all present, and in order to become the cancer, the chromosomes were lost. But we're actually finding that in the subsequent divisions of these cells, they don't have that chromosome loss anymore, and they actually return to that normal diploid state, which means they have 46 chromosomes. That's the normal number of chromosomes that patients have. So the next thing we did is we wanted to look at this in patients, and so we took patients that had an indolent form of chromophobe RCC, meaning it's not very aggressive, and we stained that for TRP13, and the TRP13 is not being expressed. You can see over here, this has a lot of brown here. This is our cell line, our model, that is a sarcomatoid variant cell line, and you can see that this has a lot of brown in it, and same thing with our PDX tissue, that patient-derived xenograft also has a lot of brown. So that means that TRP13 is very highly expressed in this. So this is a picture of mitosis, that cell division. And so this is a picture of normal mitosis here. You can see this purple here. This is the DNA, and then they're attached to this bright pink, which are microtubules. And these are lining up perfectly, and that's exactly what mitosis should look like in a normal cell. But we're actually seeing in this sarcomatoid variant that this is kind of forming what we call multipolar mitosis, meaning it's not just happening at like the North Pole and the South Pole. It's happening at the North Pole, the South Pole, and the West Pole. And you're getting this kind of aberrant, incorrect mitosis, and so it's allowing these chromosomes to do something strange and is probably causing these results, okay? So then we wanted to find out what happens when we get rid of that TRP13. So we actually have a small molecule inhibitor of TRP13. It's called DCZ0415. And we basically put our UOK276 cells into the flank of the mouse, and we treated them with this small molecule inhibitor and compared that to vehicle, which means that it's kind of like a saline variant. It's like a placebo, essentially. And so we found that if you treat the mice for 21 days with this drug, then they actually had a decreased rate of tumor growth. And then when you take out the tumors and weigh them, the weight of the mouse tumors that were treated with the drug were much lower than the weight of the tumor in the placebo form. Okay, so then we went and actually knocked down the TRP13 gene. And so you can see this is what we call a Western blot, and you can see here this is the TRP13, and so when the band is really bright, that means it has a lot of TRP13, and when the band is not very dark, it means it doesn't have a lot of TRP13. So you can see this is our TRP13 that we knocked out, and then this is our TRP13 that we just did, we use as our control, okay? And so we basically compared this cell line, these cells to these cells, and we did a bunch of assays on them. So we looked at cell proliferation, at invasion, and migration. And so this is our invasion assay, this is our migration assay, and this is our proliferation assay. And it shows that TRP13 knockdown actually had decrease in both proliferation, migration, and invasion. So in conclusion, we found that there's more TRP13 protein present in our TP53 mutant sarcomatoid chromophobe renal cell carcinoma cells. And we think that this protein actually plays an important role in the invasion and metastasis of this aggressive chromophobe RCC variant. And so we hope to investigate this further as a potential drug target. So the reason this is exciting is because this is a potential targeted therapy specifically for chromophobe RCC, and we don't have any treatments that are specific for chromophobe, and chromophobe isn't really responsive to the other drugs that we're using to treat other types of renal cell carcinoma. And so, you know, potentially having something like this in the pipeline is something to get excited about, and we're getting that much closer to cracking the biology so that we can make better therapeutics for this cancer.